In this video, we're going to have the look at basics of the cell membrane. We're going to look at the phospholipids, which make up most of the cell membrane, and then we're going to look at about how the phospholipids line up with each other. And then the next thing we're going to look at is all the stuff that's embedded and sticking out of the cell membrane. Talk a little bit about what it does. So let's have a look at phospholipids. So when we're looking at phospholipids, these are a really important molecule, and they're a really interesting one because of their structure. That when we look at a phospholipid, now this whole thing right here is a phospholipid. Now, the phospholipid has two major sections right up here, and the head and then the tails down here. Now, there are some key differences, and this is what makes a phospholipid extremely important and really essential to cell membranes. So let's start up here. Now, we're not going to go through all the chemical structure up here, but you can see why it's called a phospholipid. We have a phosphate group that's buried right there. That's the phosphate portion of what we're going to call the head. There's also a glycerol backbone, and glycerol has the 3-carbon hydrogen structure here. And then coming off that, we have two tails. And these tails are fatty acids in this case. Now let's have a look. So up here, this head, which once again is a phosphate group and a glycerol, we're going to call this the hydrophilic head. Now let's break down that term, hydrophilic. It means water loving. This part of the molecule really wants to be surrounded, embedded in water. Now that's quite different than the two tails that are sticking out down here, because these two tails are the hydrophobic tails. And these do not want to be near water. They absolutely hate being near water and want to be away. Now this dynamics, this construction of this molecule is what allows phospholipids to self-assemble into a phospholipid bilayer. And let's have a look at that. So here we can see the phospholipid bilayer. Now here's our individual phospholipids. You can see with the tails. And what this does is they spontaneously, we don't need any extra energy, we don't need any enzymes to force them in there. They will just naturally go in this position because, of course, up here is the extracellular space. And of course, this is a space full of water. Inside here is the intracellular space, basically the inside of the cell. And of course, what's inside the cell? Mostly water. So we have this dynamic where we have basically these areas where we have lots of water up here, lots of water down there, and what happens is the hydrophilic heads will arrange themselves to be facing towards water. And the hydrophobic tails will move towards the inside where they are shielded from the water. And this is beautiful. This automatically gives us our lipid bilayer. Now, that being said, that is not a complete cell membrane yet because this is kind of liquidy. And we have to put some stuff inside there. So let's have a look at what's next. So here's a diagram that starts to show some of the things that we're going to come across. So we can see our phospholipids right there. We have various phospholipids in our bilayer, and you can see, that makes up most of it. Now, let's look at some of the things that are embedded within here. And one of these may be surprise. It's cholesterol molecules. And let's zoom in. You can see there's cholesterol there. There's cholesterol there. You can tell cholesterol, you can tell what it is largely because, a good cheat sheet is it's these multiple rings that are attached together. Now, why is the cholesterol here in the cell membrane? What it does is it helps to stabilize the cell membrane. And we don't want a cell membrane that's too liquidy or also just fall apart on us. So by inserting and incorporating some of this cholesterol, it helps to stabilize the membrane till just about the perfect consistency, which is great. Now, you also notice we got all these things that look like they're blue. They're not actually blue in real life, but these are the things that are inserted in here. And we're really looking at largely things like proteins. So there's a whole number of proteins that we find associated with a cell membrane. Now let's start to give them some names. So if you look carefully, you can see some proteins are just on the periphery, either just on the inside or just on the outside surface of the cell membrane. They don't go all the way through. So here's a good example right here. This one right here, it doesn't go all the way through. It's just right here. So we're going to call this a peripheral membrane protein. And here's another one right there. Okay, so that's one group of proteins if we want to kind of start lumping them and putting them in groups. The other one is the integral proteins. And these basically are embedded in the phospholipid bilayer and often go from one side to the next. Now, that being said, what are some of these proteins doing? Well, some of them are enzymes, and some of them are about transport, to transport things across the cell membrane. So not surprisingly, some of these proteins form channels. And these are examples of channel proteins that can control what can come and what can go from inside the cell. Because remember, the most important thing about a cell membrane is it gives you some control as to your internal environment within that cell. It allows you to conduct cellular homeostasis inside there. So if I need something, it can come in, but I got to be really careful about what I regulate here. So we have our peripheral proteins, we have our integral ones, and some of the integral ones, which go all the way through, are channel proteins. Sometimes we call them trans proteins because they transit from one side to the next. And then if we start to look, we can start to see that we have some of these things sticking out, little fuzzy bits. Now, what are these? Let's have a look. Let's go over here and look at this one right here. And this is an example of a glycolipid. Now, always look at the names. The names are the cheat sheet. Glyco for sugar, carbohydrates. Lipid for, of course, lipids, fats. And this is an example of this conjugate here, where two things have come together to serve a purpose. And in this case, what I have is basically a carbohydrate that's attached to the head of a phospholipid. So it's a lipid with a carbohydrate attachment. So that's one category. That's the glycolipids. But if we look over here, we can see a couple examples of this. Here's a protein. There's a protein that's embedded in the membrane, and we can see that we have a carbohydrate chain coming off that protein. And in this case, it's not a glycolipid, but because it's attached to a protein, we're going to call it a glycoprotein. So that's a protein with carbohydrate attached. Now, one of the main takeaway messages is the outside surface of cells is fuzzy. Now, what are all these fuzzy bits doing? Well, that's another little mini lecture that we should do, but just give you an idea. Some of them help with attachment to the tissue around it. Some of them help to signal to the immune system what I am and interacts with it. So there's a number of things they do, but the main takeaway about all this stuff on the outside is cells are fuzzy on the outside, and the fuzziness has a number of jobs. Well, that's a quick introduction to cell membranes. And remember, your cell membranes are not absolutely solid. They do have a little bit of movement within them. It's stabilized by things like cholesterol, and we often call this the fluid mosaic model of the cell membrane, because that allows us to recognize that this membrane is somewhat fluid. Mosaic means it's made of a number of things, and some things can actually float. So some of these proteins actually float through here like icebergs. Now, some of them are attached, and it's something we haven't talked about in this video, but underneath here is something called the cytoskeleton within the cell, and some of these proteins will be attached to the cytoskeleton, so they're not allowed to float through. So some float around like an iceberg, some are attached. They all have different jobs, but that's a different video. Just want to give you a quick introduction. If this has been handy, found it useful, hit the subscribe button. Let me know what you want me to cover, or if you have any questions. Thanks for listening. We'll talk to you soon.